Hello, today we're starting Target 4C, Drawing Conclusions. This is lesson 4.8, Inference for Experiments. Our learning outcomes for today's lesson are to outline an experiment that uses a completely randomized design, explain the concept of statistical significance in the context of an experiment, and to use simulations to determine if the difference between two means or two proportions in an experiment is significant. So we're going to go back to our putting example. And if you remember in our previous lesson, my statistics class decided to design an experiment to determine whether anchored putting, long putters, was better than traditional putting, short putters. So here is a diagram that details how we conducted our experiment. So we had 20 randomly selected seniors from our population of seniors at Burroughs High School, and we randomly assigned them to two groups. So we either use a random number generator or we use slips of paper to split these seniors into two groups. The first group contained 10 seniors. The second group contained 10 seniors. And we assigned the first treatment to the first group. The first treatment was using a long putter. The second treatment went to the second group and they used a short putter. And then to determine the results from our experiment, we decided to compare the mean difference distance from the hole. So comparing the average distance the ball got to the hole after we putted. So here are the results. We have our data for the long putter. We have our data for the short putter. We're going to go ahead and compare these values by finding the average difference, or the average for each putter, and then we'll find the difference. So the average for the long putter was 2.8. The average for the short putter was 5.7, and the difference in those averages, or those means, was 2.9 centimeters. So what we want to do now is try to decide, was this 2.9 something that's large enough for us to say that the long putter is better? Or is it possible that we just were really lucky and we did better with our long putter just by chance? So we're going to investigate our results a little bit further to see if we can make any conclusions based on this difference in means. So in order to decide if the results are statistically significant, we'll use Statlet to simulate many experiments to see how often this result occurs strictly by chance, assuming the type of putter does not matter. So right now we're going to look at these results and say, okay, if we assume that the putter doesn't matter, then we're going to see how often this 2.9 comes up in our simulation. So to do that, we're going to go ahead and go to Statlet, enter our data. This is quantitative data. There is two groups. We have the long putter group and the short putter group. And we're going to go ahead and scroll down on our screen and go to the bottom where it says to perform inference. So we are trying to simulate a difference in two means. So Statlet is going to produce results that represent a difference in two means for our experiment. So we produced 50 additional results, and that means that every dot on here represents one experiment where we summarize that with the difference in means. So we can see that a lot of our differences were below that 2.9 value that we produced in our experiment. All right, so now what we're going to do is we're going to see how many values are equal to that 2.9 or above. We want to see the dots, how much of our simulated results are greater than that 2.9, to see if we just received that by chance or if it's because of the treatment. So there are no dots greater than or equal to 2.9, so that would be 0%. So we see here is 2.5, this is probably about 2.7. So we don't have anything that's above that 2.9 centimeters for that difference in means. So what does that mean? Do we think the difference in means we found from our experiment is due to the treatments, or has it occurred purely by chance? So we can say that no, we don't think that this happened by chance because we were able to look at simulated results and we found that that was really unlikely to produce that 2.9 difference just by chance. So we we um, we found it was unlikely to get a difference greater than or equal to that 2.9 again purely by chance. So what that means is that it was caused by the putter that was used in the experiment. If it didn't happen by chance, then if we have a well-designed experiment, it happened because of one of our treatments. All right, so some other big ideas from this lesson. In a completely randomized design, the experimental units are assigned treatments completely by chance. So that is, we want to make sure when we are assigning treatment groups, we are performing a random assignment to split our sample into two groups and then to assign a treatment using either a random number generator or using slips of paper that have been randomized. So another thing that we want to remember when we're performing a completely randomized design is that our treatment groups should be equal, if not very close to equal. So it helps to have groups that are almost the same size or the same size. It just makes our results a little bit more reliable. And there also should be some sort of response variable and a measurable outcome. So in our experiment, we measured the distance the ball got to the hole in our putting. And that was, we were able to compare the results for each group and then again compare if the treatments were affecting those results. When an observed difference in a response between the groups in an experiment is too large to be explained by chance variation in the random assignment, we say that the results are statistically significant. So what that means is that since we were not able to say that our results of a long putter, so if it occurred by chance variation, so for some reason we had all the people who are experienced golfers do really well, and that's why our experiment outcome was the way it was. So that would be like a chance variation. So it just happened to have groups that produced a result that wasn't because of our treatment. So since we were not able to explain the results by chance variation, then we know that the difference was too large to occur simply by chance. So that again means it's statistically significant. So when we're looking at our simulation results, when we looked at our experiment, we didn't have any results above the 2.9. Now that won't always happen, but for results to be statistically significant, a good cutoff is about that 5%. So if you have 5% or less results that are greater than your value, then you can say that your results are statistically significant. All right, so next up, go ahead and find the 4.8 application on Google Classroom. Try to work through the problems on your own, and then we'll talk about the answers in the next video. I will see you then.